Okay, welcome back to Anatomy and Physiology. Today I'd like to talk about the concept of membrane potential and how that can lead to an action potential. And so we'll start with here the concept of a membrane potential. And a membrane potential is a distribution of charge across a cell membrane, and it's usually measured in millivolts. So the standard is usually to compare the inside of the cell relative to the outside of the cell. So the membrane potential is the value representing the charge on the intracellular side of the membrane, based solely on the aspect of the outside of the cell being zero. And so what we have here is, again, that reference electrode will kind of, there's our zero or a reference point on the extracellular space, in the extracellular space here. So in the outside of the cell. And then inside of the cell, in the cytosol, we have usually somewhere in the range of negative 60 to negative 90 millivolts difference between the two. And so you might be asking yourselves, like, what is it that's responsible for that charge? And so there's a unequal distribution of primarily sodium and potassium inside and outside of the cell. As you can see here, there's a high concentration of sodium on the outside of the cell or in the extracellular space. There's a high concentration of potassium inside the cell. And then you can see here that these are negative aminos. So these are negatively charged aminos here that kind of increase the negative weight here close to the cell. And so the primary example or the primary mechanism responsible for this distribution of sodium and potassium is the sodium potassium pump. Now, if you recall here, the sodium potassium pump is responsible for facilitating, here it is, for facilitating the release of 3 sodium into the extracellular fluid. So 3 sodium plus, we'll put plus 3. And then 2 potassium into the cytoplasm or the cytosol itself. So the sodium potassium pump responsible for maintaining that concentration gradient of more sodium outside of the cell, more potassium inside of the cell. But then you can see that there's an unequal distribution here, that ultimately there's going to be a negative plus 1 charge outside of the cell relative to the inside of the cell there. So the sodium potassium pump in irritable muscles, or muscles that respond to stimulus that we'll see and create an action potential, they rely heavily. They use a lot of their energy on this here sodium potassium pump. And so there we have kind of that concept of membrane potential, usually somewhere in the range of negative 60 to negative 90 relative to the outside of the cell. That inside the recording electrode gives us a negative charge here again. Let's just say negative 70 is the charge on the inside of the cell. And so this is that diagram of the resting membrane potential. You can see that, again, we can have depicted here more sodium ions. That's this one here. More sodium ions outside of the cell. So the resting membrane potential is created by a high concentration gradient of sodium outside of the cell, a high concentration gradient of potassium inside of the cell, as well as these negatively charged proteins that float around on the intracellular space. There's the sodium potassium transporter, the sodium potassium pump here. So this is the example that's being given right there. And so all this leads to this negative charge, this steady state. So at normal resting state, you've got that somewhere in the range of negative 60 to negative 90. And resting membrane describes that steady state of the cell. While it is a dynamic process, it's balanced by a number of these different types of ion pumps. So you can have, let's see, while we're here, you can have different types of pumps, whether they're passive or leakage pumps. So these are ones that just kind of allow the ions to float towards their concentration gradient. You can have voltage gated channels. So now this is in response to the membrane potential. So if that membrane potential changes, you can have these voltage gated channels start to open to respond to that. Mechanically gated channels. So we see this in some of the cells that help to respond to other stretch, pressure. Mechanically gated have these channels that would allow ions to float in if there were a change in the membrane that was physically a physical change. And then the last one here is a ligand gated. So this is one that responds to a change in which a ligand, a substance, right, a neurotransmitter in this case, binds to the receptor, causing the receptor to open. And so in the case of like skeletal muscle and even like nerves, the communication is stimulated here. The communication is received by ligand-gated channels. So a neurotransmitter, usually acetylcholine in the case of a muscle contraction, binds to a receptor on the muscle. And as a result, that's where we get a change in that concentration gradient, and as a result you can start to then stimulate those voltage gated channels, right? So those ligand gated channels can start to initiate a little bit of a change in concentration gradient by having a neurotransmitter bind to the receptor. Then the receptor, the protein will open up, allowing the ions to kind of rush in, and as a result, if the change is great enough, you can get these voltage-gated channels to open. And so again, the concept of membrane potential is that there's a slight charge across these cell membranes. The primary response, the primary mechanism responsible for it, the singular primary mechanism is for maintaining that membrane potential is the sodium potassium pump. Right, remember that sodium potassium pump, 3 sodium into the extracellular space, 2 potassium into the intracellular space here. But then again, ultimately, there are a few other cations that are responsible for changing that concentration gradient, calcium being one. Chloride is an anion that we'd see hit in the extracellular space in high concentration. And stay tuned, because what we'll talk about in another section is how that resting membrane potential can change and ultimately, see if I can find it here, ultimately result in a electrical current here, right? It results in a electrical current across the cell membrane, and this is how muscle cells communicate. This is how nerve cells communicate with a action potential. Electrical current referred to as an action potential. Let's type it up there for you guys, nice and big and bold. And there's the term. Stay tuned. We'll find out about an action potential in the next sequence.